Yes.
Okay.
Good afternoon, everybody, and welcome to our DVC's talk. Personalized medicine has been something that has been very much science fiction. I remember that in 2018, Helen Malherbe, who was our postdoc at the time, and I were invited to the WHO meeting on rolling out personalized medicine in LMICs. And everything just seems so far-fetched. And even now, going into hospitals and talking to my genetics colleagues, personalized medicine just does not seem to be anything that we can look at. And yes, there are no budgets in the Department of Health for this right now, but personalized medicine has arrived. And it's in the private sector, and that's where it's going to be tried, and that's where we're going to test it. And ultimately, that should be informing our patients in the hospitals. George and I have known each other for a very long time. We started off both in a very different career, and both ended up coincidentally in genetics. And we were very surprised to meet up again at an SASHG conference some years ago, where George was working at UCT under Raj Ramesar. But interestingly, George was part of that whole big thing at the end of the 90s, if you remember the excitement around the Human Genome Project. And George was part of doing a tiny little bit of all that was put together to make up the Human Genome Project. So George has now started a company with some colleagues. He's still associated with UCT, and he is offering personalized medicine, but I've asked him to talk to us, to speak to us about where personalized medicine is right now, that we who can afford it can access it, and maybe start thinking about how we can make it possible for our patients in the hospital. So without taking up too much of your speaking time, George, I'm going to hand over to you. Thank you again for making the time to come and speak to us.
Colleen, thank you for the very nice introduction. It's actually great to be here, and it's great. Thank you for this opportunity. I think you're absolutely right. You know, when I was working on the Human Genome Project, in fact, when I graduated, even many years before that, having a copy of the genome was the dream, and all the things we were going to be able to do with it were, you know, that was going to be the magic stuff. And it took a long time to get to the point where we actually realized any of these things were coming to pass. You know, the first gene therapy was a bit iffy, some really terrible setbacks, etc., etc. But we are at the point now where precision medicine, you know, genetic directed medicine aimed at an individual patient, is actually becoming a reality. And I will primarily be talking to you about pharmacogenomics. I think it's not the only area where precision medicine is going to make the most massive impact, but I think it's an area that's now live and that actually has measurable outcomes that we can actually take advantage of. So to proceed, basically, I just want to talk about the development of pharmacogenomics, where it's going, and where we think as a company we're able to actually lift it just a little bit to the next level, to actually take it from being too one-dimensional. I'm just doing this thing where we tell you the outline, tell you what I'm going to say. I'm just going to make some definitions, give you a terribly brief history of PGX. I'm going to discuss the rules of engagement, because we can't treat pharmacogenomics as if it's different to the rest of genetics. It's real genetics. It has real impact, and it makes a real difference in people's lives. And so we can't just treat it as some sort of consumer thing that's not actually science. And then I'll give you some practical examples of how we see it going, the results that we feel you can get now with the knowledge that we have at hand, and then see if we can move it on from just being genes and drugs. So, and then maybe surprise at the end, something that I think is extremely special. So you'll have to wait for that. Please don't leave before the end. I think the best bit's there. So definition: pharmacogenetics, generally speaking, relates to the function of a single gene and its interaction with a particular xenobiotic agent, a drug or a chemical of some kind. Pharmacogenomics is sort of the evolutionary step up from that, where we now have knowledge of more changes across the genome and more drugs and more changes affecting one drug or affecting different drugs at the same time. And the truth is that the terms are pretty much interchangeable unless you're being extremely pedantic. And mostly now everybody uses the term pharmacogenomics, and we use the abbreviation PGX. So I will try and say PGX in case I stumble over pharmacogenomics. So start with a really, really brief history. If anybody Googled it, you would have come up with a better history than mine. But I picked the bits that interest me. The first time we see a description of the activity or the action of some sort of xenobiotic agent and its influence in people is around about 510 BC. Pythagoras described favism, patients getting sick when they eat folio beans. And he pointed out that it seems clearly to pick people. Some people get it and some people don't. And he was not able to kind of make the link to the fact that it might be inherited. But we do know, and we know clearly that it's now known to be caused by G6PD deficiency, and is actually fairly common in the Mediterranean populations, which are the populations that Pythagoras would have been exposed to in his research. The term pharmacogenetics itself was actually coined in 1959 by Friedrich Vogel, where he used the term to describe interactions in genetics potentially causing adverse drug reactions. Now, it's a great place to start, but actually we know that pharmacogenetics doesn't only mean adverse drug reactions. It may mean drug doesn't do what it does it too fast, does it too slowly, or you need to just modify the drug in some way to get it to work correctly. So it's not only an adverse drug reaction thing. It is sometimes nice for a patient to know, as I had with a friend of mine who was having to have chemotherapy, that when you have chemo, your genes tell me that you will respond in an average way to all of them. You're not going to get extraordinarily sick or react badly or have adverse side effects. We're not expecting any of those because your genes are telling us that you're in a good place. So what we're doing is move that term on a little bit from what it was. Where am I? Okay. Then in the 50s and 60s, I picked that because that was kind of the time when people were doing all sorts of cool experiments about discovering how genes, or what they then came to describe as genes, were being inherited, how segregation happened in disorders, etc. And they started actually describing some of the common biochemical changes that were known to be inherited, such as G6PD deficiency, which is hemolytic anemia, and prolonged apnea, which we're calling malignant hypothermia. And we now know two genes, CACNA1S and RYR1, have polymorphisms that actually predispose you to these. So we can avoid all of the risk associated simply by examining those polymorphisms and getting to the underlying causes. In 1968, a researcher called Vessel did some very cool work, which at the time was very sexy. Everyone was looking at twins, because we know that if you take identical twins, they have the same genomes. And if you take fraternal twins, they're brothers and sisters. And so you have the ability to view 100% the same genome in two people, or only 50% the same genome in two people. And he clearly showed a difference in the way that these identical twins were processing drugs the same more often than fraternal twins, than brothers and sisters, which was in the old days one of the best measures of how much genetics had to do with a characteristic, is looking at twin studies. So then, if we come then to the 21st century, the post-genomic era, we see that we have now information on masses of information on changes that have been firmly linked with phenotypes. In other words, a reduction in the activity of an enzyme, in the processing of a drug, etc. In 2006, Metelsky made an observation which I thought was actually really cool, and if you don't mind, I'm going to read it to you. He said that the specific genes that determine a pharmacogenetic response should be tested without regard to genetic ancestry, since the relevant traits usually exist in all populations except at different frequencies. So what he's saying, and which we believe to be really true, is that if you have a null mutation in a gene, it doesn't matter what population group you're in, you have a null mutation. And the effect of that will translate, whether you're Asian, Indian, European, or African, into the same processing deficiency, if that's actually the consequence, as it would with every other race. So we need to actually bear that in mind. There is a massive divide, we know, between all of the really good work and detailed work that's been done in the European, American, and Northern populations, compared to the lack of work done in the African populations. However, a mutation is still a mutation, has the same effect. And what we want to do is say, well, we'll take that, but then we need to add on to it. In the Post-Human Genome Project, what we really have is this incredible flood of data. So we got the human genome and we started being able to pick up variations in basically every gene that did everything. And acquiring all of this knowledge has led to a little bit of the problem, which I will discuss in a few minutes, is that you can't just go and say, oh, look, here's a change and it probably makes a gene defective and therefore you're not going to metabolize aspirin. Thank you very much. Well, no, it's not that simple, but we're going to, hopefully we'll clarify that just a little bit in a minute or so. The tools that we have available now, really, we are able to use microarrays, and we can analyze literally hundreds of thousands, millions of SNPs, certainly a million with pretty good reliability, which means that if I can plot a million changes, put them into one experiment, and test them on an individual or on 24 or 96 individuals at a time, if I wish to. So what that says is, I'm not going to go on a voyage of discovery. I'm going to take things that I know. I'm going to take pharmacogenomic markers that I know have a measurable or effect or an outcome, and I can test them in really significant numbers across a large number of people quickly and get good results, as opposed to sequencing people or sequencing genes in people to find out what changes they may carry that may influence the metabolism of some gene downstream. We're not necessarily sure that that's actually right now the sharpest way to do it, but it is possible. But what we use is microarrays with the access to incredibly large numbers of SNPs. So I mentioned right at the beginning that we don't want to lump pharmacogenomics into the consumer kind of based genetic testing. It's real hard science that actually can make a big difference in patients' lives, and a big difference in doctors' lives if they know that they actually can avoid the admittedly rare situations where they may be treating a patient with a wrong drug. So we know that we have data overload. As I said, when we can screen 100,000 or 900,000 SNPs in one go, we're going to get just too much information to do something with. But actually, when we make proper rules of engagement and we say we will set parameters whereby we trust the data enough to say, I can reliably go to a patient and say, yes, this effect in you will be X because the data is well supported and scientifically grounded. So that comes really to regulation and harmonization, to know that if you're doing a pharmacogenomics test here or in the UK or in the States, we will be looking at the same sets of markers, the same changes, and we will be saying the same things about them. And this adds really to the reliability and credibility. You know, we don't want to be saying, please eat more onions. Not that there's anything wrong with onions, but this is not the way that we want to see the data that we're handling being used. We want to see this can make a difference in a patient's life, either in the drugs they're taking, going to take, or not to take, as the case may be. And there's a scientific base for it. So in order to do that, I need to discuss this thing called levels of evidence. So around the world, there are a number of bodies, consortia or bodies responsible, and things like the Clinical Pharmacogenomics Implementation Consortium, largely based in the States, but it's very much a multinational. The Royal Dutch Association, which is also the pharmacogenomics, the Canadian Pharmacogenomics Network Drug Safety, and the French Network for Pharmacogenomics. Now, they all publish data, they all validate data, they all read all the published papers, or at least as many as they can, and they try to create a unified impression of where we're going with this massive amount of data that's being published all the time in different types of trials, in different types of studies. And they create guidelines that can then be used to actually make decisions on interpretation of pharmacogenomics. And they are hosted mostly by, which is easy to find, in PharmGKB. So when all else fails, type in PharmGKB, go there and type in the name of the drug. And if you're prepared to be swamped by a lot of info, you're in the right place. But what it does is it tells us that it's not just me, George, going, oh, this drug and this gene, they work together really well. No, we can't do that. We need to actually lift the level beyond that. So in PharmGKB, they publish, and this is actually a picture that I have nicked from their website, so you can see I have to attribute it down at the bottom. They actually rate any interaction that they cite, be it a SNP versus a drug, or a gene activity level versus a drug, according to the levels of evidence. And I hope you can see my cursor. The level 1 is the highest level, and 1A means it has been backed by multiple studies in large populations across different parts of the world, and they all support the same finding. So when you get right down to level 4, just at the opposite end of the spectrum, that's the kind of, George said he tried it on 5 people and they all felt better. So we're not going to publish to anybody, or certainly not to any patient, stuff on a level 4. We're going to say what we can use is level 1A and B and level 2A and B. And those tend to be, in reliable laboratories, the only levels you can use. What it does do, though, is it sharply reduces the number of interactions or relations that you can use. And this is just shown. There are currently in PharmGKB 289 drug marker interactions at level 1A, only 16 at 1B, and I think it's probably because the distinction between them is fairly minor. 44 at level 2A, 24 at level 2B, and then all the bridesmaids but never the bride. These are all the papers that are published that are pretty good papers, but are not yet backed up by large enough studies, or where the evidence sometimes goes one way and sometimes goes another way. And obviously, you can't tell a patient you're going to not be able to metabolize this drug when actually half of the paper said you metabolize it perfectly well. What we also know is that coming out of this, out of those 4,000 interactions are the ones that are going to be bopped up into level 2 and level 1 as more research is done on them, as we get a clearer picture of what they do, and as more supporting evidence is actually accumulated for them. Down at level 4, a small number, 267, and that's really because, in fact, mostly it would be there because the research is just not of the right standard to actually push them up into level 3. And obviously this assignment of levels is volatile. So I can't take a list from 3 years ago or 2 years ago even. The list gets updated regularly, and interactions get moved from one level to another. Sometimes they get moved down, but mostly they get moved up as more data has been acquired on them. So just to give you some numbers, when we examine data, we use an algorithm to process the pharmacogenomic interactions. We use a thing called a gene concept, and that's because in some cases, a gene may act on one set of drugs, and different mutations in the same gene may act on different drugs or may act differently. And so what we would say is, if the drug AB1 sometimes inhibits these drugs, some mutations, and other times doesn't, then we would call it as 2 separate events. And so there are 47 of these gene concepts which directly affect 77 drugs or drug families, because frequently the drugs really all behave the same in a family if they're a type of drug. And we, in order to get that data, we extract data from 120 SNPs, which are all level 1 and 2, mostly level 1A and 1B SNPs. So just to show you some examples, so really you should ask, what will a modern pharmacogenomics report contain? it needs obviously to say what we looked at. So it tells you a list of all the variants, either specifically by naming the SNP, or telling you which gene it's in, or translating it into a diplotype, which is sort of an activity-based interpretation. And it needs to tell you what they do. So in other words, does this make the gene upregulated, more active, less active, null? And that's important because that's actually where the interpretation of the activity comes in. And then what is the consequence of that? You carry the variant. What does the variant do to the metabolism by that enzyme, and how does that impact the drugs? So I'm showing here an extract from one of our reports, which just shows the gene names. So you can see the name of the gene, and in this case, ABCG2. It gives you a result GT, which tells me there's actually a single SNP that's been looked at. And if you're a GT, as this patient is, so heterozygous, they have a G allele and a T allele, in that there is a decreased function. So in all the things that we know to be assigned to ABCG2, they will be affected by it having a decreased function. A little way further over here, you can see under the cytochromes, the CYPs, the cytochrome P450 types, you can see that actually they have a different allele nomenclature. They're using a star allele. Now, in the case of CYP2D6, which is one of the most common drug metabolizing enzymes, we currently use 20 SNPs just to make sure that we capture the typing of CYP2D6 accurately. So in some cases, we're only using one SNP if it's been well described and based only on one SNP in a gene. But in other cases, and in cases like CYP2D6, where the activity, where it is very, very widely implicated in metabolism of up to, I think it's 30 to 40% of drugs pass through or are handled by CYP2D6 at some stage in their lives. And you can see that this individual is a star 2A, star 4A, and which says that they're an intermediate metabolizer. So it's showing you the 2 points. I'll tell you the typing, and I'll tell you the consequence in terms of what it means for the activity of that particular gene product. And then to the second part of or the third part of what I said before is, so what does this mean for drugs? So this is just an excerpt. There are many pages of this usually. We break the drugs down into the categories of drugs that they are, and then we say, use with standard precautions. In other words, there's nothing that we can see in your genetic predisposition that says this drug should not work as predicted. Use with caution, the yellow column, so it's a little bit like traffic light. The yellow column says use with caution, but it should be okay if you're paying attention. Change recommended. And you can see that in this particular patient's case, carbamazepine, because the patient is HLA A3101, star 3101, they are at an increased risk, and you should try not to give them carbamazepine. So what we do is we recommend, because obviously the clinician makes the decisions, but if we can give information to say, you can actually shift away from the things in red and look into the agents in the green column, simply because that means the patient will be getting the same type of drug, the same family of drug, but actually metabolized or processed in a way that's not being interfered with by the genome of that individual. So, I mean, that's pretty good. But what we want to be able to say, well, hang on a minute, it's not really the whole story. I mean, we know that there's more to it, right? We know, for example, that while my genetics doesn't change, the environmental factors around me do, and the drugs that I take at various stages in my life are going to be different. You know, some emergency or some catastrophe happens, and I may be taking pretty terrible medicine for quite a long time. But even just sicknesses or aches and pains or broken things can cause me to change my drugs. And the drugs themselves don't generally work in isolation. In other words, I'm not taking a drug and there is nothing else in me that could mess with it, that could upregulate it, downregulate it, bind it, release it too quickly. That's not true. We know that that is true, actually. So, and that my lifestyle changes, and we know that that's definitely the case. There was a time when even I didn't drink. And however, that frequently changes and sometimes not so true, right? But so what we're trying to do then is say, well, how do we get then to the next level? And I said From the previous slide, you may remember 47 gene concepts, 77 drugs, 120 SNPs. We now have added to that using an algorithm that you can input treatments that the patient's on, from which we have a library of 13,000 drugs. Now, admittedly, that sounds terribly impressive, but it is things like aspirin 100 milligrams, aspirin 50 milligrams, aspirin quick release, blah, blah, blah. So it's not quite 13,000 discrete drugs, but it is a large number of drugs from which you can now say what I'm giving to this patient or what I wish to give to this patient, because really the question is the same. I'm not looking at the patient, I'm looking at their genes. So whether it's a drug they're on or a drug you're proposing for them, from our point of view, it's the same effect. So we can generate a result looking at the drugs that the patient is using. We can also generate an interpretation saying, if I want to add some new medication to the patient, can you tell me how it would work? And then we have 22 lifestyle factors that have been found reliably to influence drug metabolism in some way or another. I'm not going to list the 22 because I always find them quite confusing, but they are there. So if you request a patient form, you will see the 22 lifestyle factors. And what we now get is the ability to generate a map. It's called a heat map, which shows the contributions. And I'm sorry, I'm just going to turn my head a bit here so you can see there's the 3 columns, which is genetics. So the bupropion hydrobromide right at the top there has an M. There is a medium risk, genetic risk for this patient if they're taking that. However, if you look at imipramine, there is a high risk of drug interaction. Because in other words, I've now said this patient is on a number of medications, and what the software does is looks at the genes and says, okay, the genes interact with the drugs, but then it looks at the drugs that the patient is taking and actually extrapolates, would any of these drugs interact with those? And then it also does what it calls the ACV, the anticholinergic burden. So obviously if it's pathway usage, if the drugs are all working on the same pathway, the pathway overloads and stops working or becomes faulty. In older patients, there will also be a 4th column, which is the Beers criteria, which is the geriatric treatment parameters that are set out by the American College. And it helps because they specifically assign values to certain medications that they say should be treated with caution in older patients. So we get this heat map, which now tells us the existing drugs, the genes, and any new drugs that the patient may be taking, and how they may interact with drugs in those classes. And then we get the second thing, which is a risk chart. So based on the drugs the patient is taking now, this risk chart shows me, so in this case, the patient is taking those 3 drugs, and there are 2 categories that it's showing me as having some warning. There's a drug interaction and an FDA black box. And the FDA black box warning is there because we know that standardly the FDA prescribes for certain medications a warning that everyone must read. However, we tend to turn it off because they tend to be for very extreme but rare events. So if we subtract 15 from the risk chart that's shown here, then the patient's actual risk is between 5 and 10, which is quite good. But you can see that they're drug interaction risks. There is no genetic drug risk in those 3 drugs that have been given for this patient. So this is useful because the clinician can now quickly identify possible problems, what the cause of the problem might be. In other words, is it a genetic or a drug interaction problem? And you could say, for example, in this patient, fluoxetine and clobazam probably are interacting with each other. And in fact, in the report, it will tell you that further down in the page that I didn't show on the screen. It will say that they interact in what way. And then you can go and look in that family of drugs and choose something else to try and reduce the drug risk for that particular patient. We believe that that's quite cool because we're now adding this second layer, not just looking at the genetics, in isolation, looking at it in the environment of the drugs you're taking or drugs you may be taking and lifestyle factors. But we have an opportunity really now to say, is there something else? And I think there is, and we think there is. To take it even further, because the question really arises, so I have my pharmacogenomics done today. What about next month or in 3 months' time where I have a cardiovascular event, right? What do we do then? Do you send my blood back and ask to do it again? No, that seems really unfair. So what we've created is a second chance, as it were. We've created this, which is a live report version, and you may recognize it as being pretty similar to the one I showed right at the beginning. This is just showing in a standardized, canned way, these are your genotypes, and these are the consequences for them. But you'll notice that right at the top of the report, it says it's a live report, and it has a URL and then 2 keys. Now, those 2 keys at that URL, which is not real, so don't write it down, will actually allow you to access this patient's unique results that we've generated, and will show you the lifestyle factors, will show you the drugs and the genotypes, and will actually in real time interpret them for you, meaning change my drugs, modify them, tweak them, change the dose, do whatever you want to do, and then it'll actually give you a second chance to optimize the patient's outcomes or the patient's treatment regimes based on good underlying science. When you click on the link, one of the things you get to, it's a little bit blurry, but you can see down the left-hand side, it says, it shows you about the genetics, the demographics, the lifestyle factors, the indications. You can actually add disease categories there, and the regimens. If you add disease categories, what it does is it suggests drugs that are related to that disease category when you ask it to tell you about drugs. And what I've put it on is the medication regimen. So the patient I chose for this example has a number of drugs that they're taking, and you can actually change that. You can add to it, or you can take drugs away, and in real time see the consequence in terms of what does this do for the drug gene, drug lifestyle interactions for this patient. It then takes you to, once you've made the changes, you say, please show me the report, and it regenerates that risk chart that we looked at before. And you can see that this patient has a number of risks for only 2 of the 7 drugs that they're taking. And there is a drug-drug interaction, which is shown in yellow. And then there's a drug lifestyle interaction shown in green on the bar chart on the right-hand side. Now, that is actually quite interesting because, when you hover over it, it tells you what it is. And it says this patient has told us that they take marijuana, and marijuana interferes with dexamethasone. So lifestyle change is pretty much recommended for this patient for a pretty good reason, at least. You're not just saying, I disapprove, son, stop taking it. You can actually now give a scientific reason. And that's, I think, is really important. The second thing on the left-hand side here, which is the side effects, I would suggest to any clinician who's ever facing a patient, and you would probably suggest to me, that we're never going to tell them the side effects beforehand, because then they will get all of them. However, if a patient is presenting with side effects, here it can tell you the relative contribution of the various medications to those potential side effects. So it just helps to give a picture. So if someone has experienced side effects, you can actually say, when you're using this interface, you can say, here we go. We know why, because these 2 drugs are both contributing in the same way to actually generating that one particular side effect, which I think is quite useful. And then the clinician now has the option to go almost back and forth, choosing drugs to say, how is this going to modify it? What difference will it make to the patient outcome? And in fact, this tool even has the option to recommend alternatives. So you can tell it a treatment category, and it will tell you what drugs it thinks are available. And when it lists those drugs, it actually gives you the little map of how they interact with this patient in terms of genetics, drugs, and lifestyle. So you can choose a drug that's actually going to give you a low interaction with the genes or a low interaction with the other drugs. So it really does make it possible and to really optimize it and give a very good, scientifically at least verified drug regime that should actually be as safe as possible for this patient. So, yeah. So when all is said and done, we say, how important is this? I've said before that we're not talking about a direct-to-consumer product like ancestry prediction, which I'm sure is terribly useful for some people, but the science of it is not immensely strong, and I'll probably get threatening letters from scientists who do ancestry. But the truth is that telling me I'm 2% Irish is possibly not a surprise, because that same 2% could make me Scandinavian. However, in pharmacogenomics, we come across some quite striking examples. I've just chosen two, not the most scary ones, but ones that I think are probably really useful. We know tamoxifen itself is a prodrug given to breast cancer patients. The drug itself is activated by the enzyme produced by the gene CYP2D6. If you are a poor or a null metabolizer of CYP2D6, in other words, the patient I showed you before was an intermediate metabolizer, so you're not metabolizing quite as well, then if you are taking tamoxifen and converting it into endoxifen, you're not doing it well enough. You're not doing it as well as the average. So it's possible that you could be taking the drug and not getting a therapeutic dose, which is really sad, because this drug is meant to be doing something immensely important in your body. So even intermediate metabolizers may need, the recommendation is that they may need a slightly higher, but they're very carefully worded, but still FDA approved level of the drug. Obviously, I think in these cases, they could still be used, but would require really close monitoring for side effects, and that you're reaching into the therapeutic range of the dosing. The second example is Fluorouracil, which I'm particularly fond of as an example, because I think it has a lot of ramifications for us. Even internationally now, we've seen international guidelines are saying that 4 common alleles of the gene DPYD should be examined before administering 5-FU. Now, it gets really cool here, because these SNPs, these changes, actually occur in quite a range of frequencies, 3 to 5% in Europeans, about 8% in Africans. And I was pointing out when I was discussing with Professor Aldous earlier, is that I've seen a recent publication where there are some of these changes that occur in South African Zulus at the rate of almost 12%. This is not a disease-causing gene, so it can be at a fairly high frequency. But if these patients are exposed to Fluorouracil, sadly, then they will have adverse reaction, and 12% is a really high number. In cases, out of 2 publications on the dosing of DPYD, the DPYD-based dosing of 5-FU, there is a 25 times increased risk of treatment-related mortality. That's massive. And that's only testing for the 3 SNPs that they mentioned there, the 1905, the 1679, and the 2846. I mean, that really is crazy to think that you can make such a difference, and all we have to do is look at one gene. We're only looking at DPYD. For us, this is really extremely exciting stuff. I want to point out that we will look at SNPs. In other words, we will look at the known changes with known consequences. Because as I said previously, we have to comply with the guidelines, 1A, 2A, 2B. And one observation of a bad reaction in a patient is not enough to actually make these guidelines in place. And so it leads us to the really important thing, is that we want to get involved in research. We are a diagnostic laboratory, but actually it's really important for us to say, we know that in our African context, we can find new things and we can extend what we know. And so we're looking for collaborators, we're looking for people who want to do scientific work, who may have access to patients and can say, let's set up a project, we'll try and write for some funding, do some really interesting, cool research that will benefit the indigenous populations of this country. We don't have to do research on Europeans. The research has been done already. Or Americans, or even the Asians. The research has been done at an amazing rate. But if we could then do a sequencing, not a CYP-based, we could actually sequence the gene, which we can do. We have the next generation sequencing capacity here. We can sequence the gene, find out any new changes, and see how those associate with phenotype in patients. So perhaps in the oncology wards where patients are seen to have bad reactions or adverse reactions, is that we can actually do research on them, find firstly, do they have one of the existing changes? Because it's an easy, quick test. If they don't, can we sequence their gene and try and identify the real causes underneath it all? Yeah, and that I think is pretty much it. I would like to thank the DVC who's hosting these, and I would like to thank Marianne and Francis for linking me up, and Professor Aldous for facilitating. So, and obviously everyone for listening. Thank you very much.
Oh, thanks, George, for giving us an idea of just how accessible this can be. Okay, costs are something we're just going to have to forget about while we're thinking about doing something.
Yeah.
There is a question there are a couple of questions coming up. I'm going to ask people to just unmute themselves and ask. So the person who asked about the first question, if you say you do not do DTC testing, if you just unmute yourself and let's have some discussion around this.
Sorry, Colleen, they would need to raise their hand so that I can unmute them.
Okay, could you raise your hands? The person who said, who's the first in the chat box? It's how does testing process get gatewayed? And then the next person asking about costs. While they are unmuting themselves, George, yes. I'd like to play two scenarios for you. So I'm a doctor sitting in Westville. I'm a GP, and a patient comes into me and wants this kind of testing done, or you think that this kind of testing needs to be done. What is the next step? Anati, I'm going to put your question up next.
Okay, so in terms of getting access samples to us, pretty easy. We can take, it's a blood sample, 5 mils of EDTA, or a saliva sample. We will send our testing kits to the GP. If they want one, we'll send them a saliva kit. Saliva kits are easy, no needles needed. You contact us, we will send you a patient request form, or we will send your GP a patient request form. They fill it in, the sample gets shipped to us by our couriers, and that's it. And then the nasty bit, we unfortunately have to send you an account.
Okay, I'm not talking about money right now. I'm talking about then what happens? Do I get access to this computer? Do I sit in front of the patient tapping away on the computer and say, oh, should I give you this drug? Let me try this. Yeah.
Okay, so what the GP would be required to do is, well, there's a choice. Unfortunately, we do have to talk about money. The original test that I described is called a static report. In other words, you tell us what drugs you're on, you tell us your lifestyle factors, we look at your genes, and we compile the report. That report is static and does not change. If you wish to be able to log in and query it, principally for the reason that it costs us more, we have to charge you more for that test. And that is partially because a server is being made available to do this, and that server has to be made available in the long term. It's not just, yeah, we can do it, but we can do it for the next 2 months, and then we can't guarantee it will work. We're trying to be in a position where we will guarantee that this will be a stable offering, that in a year or 3 years' time, when your life does change or may change, that it will still be available. So there is a decision that has to be made. Do I take the static version? And the static version is cheaper, significantly cheaper, because you're not asking for that. If you think about it, every time they click on something, we are doing a whole new analysis, which, although it's computational, it's still a whole new analysis that's being carried out every time. So the doctor would, in a standard report, receive from us notification that the report is ready. We would mail it to the doctor. If the doctor is not happy giving the report, and this is actually a very, very real scenario, because quite often, let's face it, if you've never given a result of this kind, you're going to be faced with sometimes 27 pages. We make our reports, we specifically write our reports so that page 1 shows you the stuff that's important, and page 2 to whatever shows you the basis that we've put that on. So if you want to know the scientific reason, read through it. If you want to know the brief answer, page 1 will tell you, are there drugs that need to be avoided for this patient? Are there lifestyle factors that need to be avoided for this patient? those will be listed. If it's a static report, that's where it ends. The doctor can get help from us or from genetic counselors, who we're very happy to recommend, since we're lucky to be based here in UCT, where they have one of the schools of genetic counseling, and we can actually get access to people who can talk to you and help you to interpret the reports, or even talk to patients if the GP himself is not, him or herself is not actually comfortable doing that. If they want the live report, long answer, sorry about this, Colleen, you wanted a short answer. If they want the live report, then the report that comes back has that link, and should things change, or should the doctor wish to query them, they can actually log on live on a computer, go and have a look at it. I would actually like to see this being done by pharmacists, probably at some stage in the future, but we are kind of bucking the trend to how doctors and pharmacists are interacting in the drug prescription arena. And that may need to be changed, but we've certainly seen with collaborators in the States that they're doing that. They're saying what they do is they send the patient for the test. The patient then goes and sits with a pharmacist, looks at their drug interactions, looks at what needs to be optimized, what needs to be changed, what should be personalized, and then take that recommendation back to the clinician and say, based on my genetics and my lifestyle and everything else, these are the best combinations of drugs that we worked out. Are you happy with that? And that is an immensely powerful, and in fact, studies done at the Mayo Clinic and oh my goodness, Corel, the CLI, Corel Life Sciences, have shown that patients, that they save money because patients are not given the wrong drugs, are not given drugs that are giving them bad reactions, etc. So yeah, did that answer your question, or did I go too far?
No, I've got a follow-up question, but I'm first going to give Unati an opportunity to ask a question, then come back to the person who asked the questions in the chat, who says they're in a very noisy environment. So we'll come back to those in the chat. Unati, would you like to pose your question?
Yes, thanks, Prof. Yes, thank you so much. It's a very interesting and exciting talk. I have two questions. So the first question I have is the drugs, so the repertoire of drugs that you have available in terms of genetic testing, in terms of how comprehensive it is. I'm just thinking in terms of public sector, ARVs, TB drugs, antihypertensives. I know you did mention the cancer drugs. And then as a lead-up question to that is the availability of the genetic testing to public sector.
Yeah.
I know earlier it was noted that your service is available more to private, but from public sector, we do send off genetics at times if it's not available within public sector to private labs. So just to find out, if you do offer that to public sector, healthcare services. Thank you.
Yes. Okay, so to look at the first part of your question, actually the list of drugs, we do publish a list of drugs, and it is possible to see, and it may well be a worthwhile experiment to measure that list of drugs against your prescribing list, which I know is in most provinces they recommend a list of drugs in the public sector that you're allowed to prescribe. We could certainly, it would be a worthwhile exercise to look at that and see what level of overlap there is. Frequently, the drugs are also, as I've said right at the beginning, drug families rather than specific drugs. But it would be really worthwhile to know, how many drugs in your sort of arena would actually be candidates for this analysis. If it's a small number, and sometimes the number is, the science is still quite young. So although we're very excited by it, we know that it's not yet comprehensive, not yet every single interaction for every single drug is known. And that will come. But we're trying to use what we can now to do some good. As addressing your question about the public sector, I think we have to realize that the costing then becomes the major issue. It is available. Obviously, if you send a sample to me, I'll test it. But the question is, how much will you be allowed to pay for that? And are you taking that away from something else, that you then not be able to give to a patient? We're hoping that by making this testing more mainstream, and I just wanted to point out, Unati, we're thinking of this as kind of the beginning of the game for us, and it is really. In the UK, they are now in the situation where from next year, the NHS in the UK will regard pharmacogenomics testing as a basic right. In other words, what's it called? What do we call here? Prescribed minimum benefit. Everyone will get it. If they need it, they will get it. They've realized when they look at the sums, when they look at the money you're wasting on the wrong medication or the consequences of adverse drug reactions, that the cost of doing a pharmacogenomics test is tiny by comparison. Even though a pharmacogenomics test may cost thousands of rands, it's still tiny compared to the very bad, potentially bad adverse drug reactions, especially when you talk at governmental level, right? Because they're talking about treating, well, we treat 50 million people because we're the NHS. So your 1 in 1,000 bad reactions ends up being a lot of people. And so the economic argument is very readily seen, and they've actually adopted it. It's now, it's happening. And my understanding is it's happening from next year, but it's happening. We will get there. We have a number of bridges to cross. You remember that what we're doing, mostly the reagents that we use here are imported, and so we're at the wrong end of the rand dollar or the rand pound or even the rand euro. And so our tests are not as cheap as you would think they could be or could be made to be. We're trying to form stronger collaborations with our upstream suppliers in saying that if we start doing this test and we want to do work, and they're very interested in this, if we say, for example, as I mentioned just before the end of my talk, if we want to do research on DPYD in Africans, that they must come to the party and give us reagents at a cost that makes it feasible for Professor Elders to write a grant, get some money and get some research done so that we can answer these questions. And they are keen to do that because they realize the importance of the work, but we unfortunately can't guarantee that beforehand. You know, it would very much be an on-the-go thing. And down the road into the public sector, whether either, well, obviously we would be prepared to do it at a lesser profit, and would our upstream suppliers be able to sell us reagents at a lesser profit to make the whole thing cheap enough that actually the provincial government could say, yes, okay, we can start offering this, because we understand that when they offer it, they will be offering it not just to 2 or 3 patients. You want for it to have an impact, to offer it to every single person who goes into an oncology unit and say, can we test you and can we start making a difference? Natia, I hope that answered your question.
Yes, you have. Thank you.
Okay, let's move on to the questions in the chat. The first 2 are from the same person. Dr. Rebella, if you say you do not do DTC testing, then how is the testing process gatewayed? By physician request? And if so, do these physicians need training? That's a question I wanted to ask specifically. By your company. I know other companies that require this in South Africa. Then he moves on to say, I'm just wondering what the costs are looking at, for example, 120 SNPs and what TATs.
Okay, so three questions.
Yeah. Can you see them? If you go into the chat, can you see those questions?
Well, I think I'd have to, if I stop presenting, I might be able to.
Yes,
but
yes, you can stop the presentation.
Just give me a second and I'll stop sharing, and that'll maybe make a difference. And let me show the chat.
George, have a look at under Q&A.
Sorry, it's under? Oh, Q&A. Under Q&A, sorry. Okay. Okay, so yeah, so the gatewaying of the testing, a really important question. I think what we don't want to do is set ourselves up to doing direct to consumer simply because we believe that the role of the clinician in this process is critical. I would hate to send somebody a report that says, don't take your medication. I mean, could you imagine? It would be a catastrophe of major note. What we want to do is say we're interacting as a provider in the healthcare provision to this patient. And so if patients hear about us and want help, we are quite happy to communicate. Our clinical director and our senior lab staff will communicate with your doctors if they need information, if they need information on what the test is about, on what needs to be done, etc., etc. In most cases, we would suggest, please contact your GP, your gynecologist, your cardiologist, whoever it is that would actually serve as the clinical support in this case. We also know that in most cases, it would be better for them to get support from somebody like a genetic counselor who's going to be able to say, this is what, this is the consequences of what this report is and the interpretation of it. However, the clinician is still the one who's going to make the decision, right? He's your primary carer. And
Jordan, I must point out the majority of the people who are in the audience are all clinicians.
Perfect. So this is the point. We don't want to take away from the point of care, because then I think we're setting ourselves up for a massive fall, because it's wrong. The care of the patient is in the hand of the clinician, and our reports will not say take them off this drug, put them on that drug. Never. We'll say there's a reason for this drug being not good in this patient. Perhaps another one is better, because it is ultimately the clinician's responsibility. The person writing the prescription is the one who takes the responsibility. And so I need to make sure that I'm helping to give them enough information that they can do it and make that decision based on the best evidence and as wisely as they can. And if we can help that, and I think we can help that by not doing direct to consumer. And we will help facilitate it by saying, if you need access to counselors, if you need our clinical director to speak to your carer or to anybody else, or you need us to help come and give talks, lectures, we're preparing a series of introductory lectures, hopefully trying to get that set up in the next few months, on how to give one of these genetic reports, then this is all stuff that we want to do. I think, addressing your point, that physicians do need training, in the sense that this is kind of an area that, unfortunately, and I don't know if the UKZN, I'll be perfectly honest, I can only speak about the UCT medical curriculum, where I've had a small amount of input. We noticed that in something like the whole curriculum, their total contact hours are in genetics.
Minimal.
What, 10, 12? It's nothing. It's nothing for what is probably becoming one of the most dominant fields in medicine. And so doctors have gone out and started practicing without the support they need to actually implement these things as they're coming available. And as you pointed out, Colleen, in 2018, which is not a very long time ago, this was pie in the sky, right? So the fact that curricula have not caught up and that clinicians are practicing without having been exposed is not surprising. It's actually, I'm surprised when I meet doctors who do have fantastic knowledge of it, because it's really only by their personal interest and their involvement that they're able to acquire that knowledge, because it was never made available to them as part of their training. So we would like to see training courses being made available, but I know most physicians are incredibly busy, and saying to them, come and spend a week's worth of your time figuring out how these things work. Maybe you'd be better off sending them to a genetic counselor who's actually spent 4 years of their time making sure that they know how to interpret these reports and give the right results, if that helps.
And just for those people here in KZN, we do have a genetics counsellor now in KZN. She's employed by Ampath, and I think she is available to all private practitioners. So then the next question was about cost.
Yes. So, do you want to know how much it costs?
Should I rather just put you in touch rather than talk on a live stream about costs?
So that's always a.
Could email me. I will then put you in touch with George.
I'm certainly happy to discuss costing. And we're trying to be flexible in terms of the model that we use for costing, because there is if we are offering the costing to a service provider themselves rather than to the final customer, it can be seen as a business-to-business transaction. It's often when involved in maybe screening programs like a staff wellness program and things like that. So I think that I can give you sort of ballpark figures, saying that at the moment, a pharmacogenomics test, a standard one, the one without anything fancy, is around about, and my boss is probably going to want to kick me, around about 3,000 Rand. It's not 20, 50 or 100,000. And it's come down. It compares pretty well to the direct-to-consumer testing pricing, I think, when you convert it into rands and shipping samples, etc., and the fact that I honestly believe that our product is advanced on what you will get if you just do a direct-to-consumer.
I think I must also mention there are pretty serious ethical issues around direct-to-consumer.
I mean, it's horrifying if you think what potentially can be given, what results can be given. You know, one of the things whenever I'm talking, so as you can see, I love talking. So whenever I'm going out to dinner and talking to people that I meet, they always say to me, "Well, can you tell me if I'm going to get Alzheimer's?" Please don't ask me that, because if there is no reason to suspect you for Alzheimer's, I have no ethical reason to test you for Alzheimer's. Right? And therefore I have no ethical basis for giving you a result. And if I mail you a result that says, yes, you are likely to die of Alzheimer's, what kind of ethics have I got? I mean, that is just completely
wasn't 23andMe shut down because of that?
Well, I think that, I think it would be, and it has to, you know, it has to be responsibly given simply because the tool is now becoming so powerful.
Yeah.
You know, we can do stuff, we can tell you stuff about yourself just by looking at your genes that is horrifying. You know, in particular cases is horrifying. You could find out you have a deadly late onset disease. And really, just to be told that out of the blue by an envelope that arrives at your door, that doesn't seem to be the right way to do it.
And I think South Africa also has national ethics codes on genetic information.
Absolutely.
So you would never, ever send a direct, anything that diagnoses anything would never go directly to the company.
Oh gosh, no. And in fact, that's our primary kind of drivers. We feel we want to distinguish ourselves. Firstly, we want to have the best level of science that we can bring to the game. And secondly, it has to be done ethically and responsibly. So,
we've reached our hour. I'm going to ask if anybody else wants to put their hand up to ask a final question.
Sorry, can I ask something? I'm looking at the chat and I'm seeing here that Vasi.
Oh, sorry, I forgot about Vasi's
Vasi,
question.
that is such a cool question. Does Vasi want to ask the question, or do you want to read it out?
I think let's just read it out. Yeah, I think it's pretty clear, a good question. Yeah.
Sorry, I just it's in the South African context. Okay, traditional medicines being used, can genomic testing be able to identify? Now, we know, for example, that the active ingredients in these traditional medicines are very seldom characterized. They are becoming so, and there's a lot of work, and I know at UCT they have a whole unit dedicated to that, and they have a professor with a very recent MRC chair in pharmacogenomics who has a massive interest in pharmacogenomics around traditional medicines. And I think that as this work is being done, as we're now going out and actually asking this question, we're going to find out that, yes, in this traditional medicine, the active ingredients are these list of components, and the genes that regulate them or interact with them are these, and yes, that knowledge is coming. However, right now, the knowledge is very sparse. Oddly, even though normal, say, Western herbal medicines are very widely used, widely prescribed, there is very little scientific work being done on what happens if I take this, St. John's wort, what's the other one? Grapefruit juice, leafy green vegetables, marijuana, alcohol. So we're not calling them the traditional medicines, but these are kind of pharmacology drugs that are taken, and they have information on them, but a whole heap of other stuff has absolutely no supporting information because no research has been done. So I suspect, and I'm hoping that Professor Dandara will keep us informed, that as he's working on the traditional medications and the responses to them, that we will be able to include that as part of what we offer, because it's extremely important. You can't, you can't pretend it's not. So sorry, Vasi, I think it was such a cool question.
As you said, we're at the beginning of this, and we've been anticipating precision medicine, personalized medicine, genetics being part of medicine since we've got the Genome Project, nearly 20 years ago.
Yeah.
This is a real 20 years. I mean, next year will be it was 2002 that we had the announcement.
Yeah, exactly.
So this is all new. It's 20 years into this, and everything is still so new. What we cannot do is just turn a blind eye and say, okay, that's for the younger people, the younger generation. It's not. We have to be there to make sure that this technology is rolled out for the good of people, that it is rolled out ethically, and that we set those standards and we begin to use it, so that with use, we get further development.
Correct. Yeah. And if I may just extend on that even a little bit to say that we know that there's a lot of information that we're still missing. Right? We know that we don't have a full list of gene-drug interactions for all the drugs that are prescribed. And we know that not every genetic interaction is understood. However, we know that a lot of stuff is understood and can make a difference. And I would rather say that while we are promoting research to actually uncover that stuff, let's use what we know. Let's offer the benefit where we can and take it forwards from there. Because that way we're offering a service to people, we're doing good, while we're encouraging research to actually uncover the next things, not wait. It seems to me that it would be a very poor excuse to wait and say, well, in 5 years' time, boy, will we be able to do a really good job. Let's not do it now. No, I don't think that's the case at all. I think we can do a really good job now, less so than we will do in 5 years' time, but we can do a really good job now.
But if we don't do it now, we'll be, in 5 years' time, we'll be where we are now.
Absolutely. Yes. We'll be starting to adopt it and struggling with exactly the same problems we're struggling with now.
Thank you, George. Thanks for taking the time. I think everybody enjoyed your presentation. If anybody would like to interact further with George, please email me and I will put you in contact with him. Thanks everybody for attending.
Thank you very much. And thank you.
We'll see you all soon. Thanks again. Bye-bye, everybody.
Thanks. Bye.